So we're going to get going. Thank you very much for being here. Welcome, welcome to Hands-On Soil Health Assessment for Farmers and Gardeners with our very own NOFA Mass Education Director and wearer of many other hats, Caro Roszell. And one of those other hats is a big part of our soil technical team. I'm probably not stating that as correctly as her title warrants, but she's a big part of our soil tech team. And we're in for a wonderful presentation, I know this. I'm going to do a couple housewarming housekeeping announcements, housewarming as well as housekeeping announcements to get us started, and then I will hand the controls over to Caro. So I first want to give thanks to our sponsors for their support of our NOFA Summer Conference overall and the work of NOFA in general as well. Please, as I'm sure many of you are familiar with a lot of these sponsors, please show them support. They support our work and we love them. Acknowledge here that we are presenting, attending, and hosting this workshop from land that was managed and inhabited before European colonization. Please take a moment to find your location on this map and honor those whose land you now occupy. I am presently broadcasting from the land of the Wampanoag tribe. And there is a link here that I will drop in the chat. I did drop it in the waiting room already. I will drop those links again in the chat. And you can utilize this map here. It is interactive and you can drill down into your region wherever you are. It's a fantastic resource. I hope you'll enjoy it. And I also will drop these links again. We have vendors for the conference that you can visit through the link. It leads you to our website, which leads you to their individual pages. Our vendors have provided specific codes or deals for you as conference attendees. So please check out the vendors and see what they're offering and enjoy their wares. And also please take part in our raffle. It supports the NOFA Summer Conference Scholarship Fund. There are some fantastic items in there from some of these vendors as well. So please, vendors and sponsors and other donors as well. So please support the scholarship fund and go home with some really nice goodies. If you are a certified crop advisor and require CEUs for your annual, I guess it's your annual certification, you can get some through attending this session through the QR reader on your phone. Here's the code right here. I will also put it up at the end. If you don't have the QR code reader, you can contact us and we will send this to you so you can get those CEUs. So now please sit back, relax, be active in the chat, send your questions as they come up. Please enjoy the workshop, and I'd like to hand it over to Caro Roszell. Thank you for being here, Caro. It's nice to see you.
Jason, hi, thanks. I'm just gonna try to maximize this right now. For some reason, it doesn't wanna let me. Great. You know you're a NOFA person when you look at that, the vendor slide and you're like, oh, I have half of those books on my shelf. And then you look at the raffle slide and you're like, oh, I have all that stuff too. All right, well, I'm really excited to be here. I'm just gonna give it one more shot to see if I can make this a little bit full screen. It worked before, but. So as Jason said, I am the NOFA Mass Education Director, and I am also the coordinator for our soil tech team. And as part of that, I am a soil health advisor for farms and gardens. And today I'm actually broadcasting to you from Simple Gifts Farm, where I worked for about 6 years. And my internet wasn't working at home, so I'm in a barn at Simple Gifts Farm right now. But I'm really excited to talk to you today about hands-on soil health assessment for farmers and gardeners. I did want to just get a quick sense. So first of all, can everybody see this presentation all right? All right, good. I just first wanted to get a sense for why you're all interested. So if anyone wants to kind of jump in and let me know, feel free to go off mute. Just share a little bit about what brings you to this workshop right now. Or drop it in the chat if you're more comfortable with that.
I'd like to say something. I always enjoy soil talks. I think it's sort of something that you continually learn. And I know you've done a lot of work on soils, and anything you might say about some of the work you found about the survey you and Jack were doing in Massachusetts on carbon and so forth would also be interesting.
Great. It looks like Helen is interested because she's in a group of soil stewards and we all want to know a little bit more. Michael's interested in nutrient density. That's great. Having more life in your soil is going to improve the nutrient density of your food. Karen's interested in carbon sequestration and improving her garden. Megan is a farmer hoping to learn more about how to understand soil test results instead of it seeming like a mystical prophecy. Awesome, cool. Well, I can help you with some of that. We're not really going to talk too much about soil lab results today. What I'm going to talk to you about today is direct observation. Jason, would you mind just grabbing the folder with the 2 items in it specifically so people, and just dropping that in the chat so people can find that more easily? Okay. So Jason's going to share a link to a folder that has 2 things in it. The first is the technical protocols. These were first developed by Jack Kittredge, and then I revised them over the past winter because NRCS led in a process led by Brandon Smith and Jeff Herrick and a few other people at NRCS. Revised, they did a big research project and looked into all the different ways of evaluating soil health, both on the lab side and the field side, and released a new sort of a technical note about it and a new set of in-field soil health assessment protocols. So I revised the protocols over the winter. So what you're going to receive in that folder is the technical protocols, which are newly revised this winter. The reason we revise them is to actually make it possible for our technical consultants using those protocols to look at the same assessment criteria that NRCS is looking at. And then the other item in that folder is the grower's manual. And the grower's manual is just a little bit of a simpler set of protocols. They're very similar. The data sheets are a little bit simpler. There's not as much jargon in them. So the grower's protocols are maybe a place to start if you're new to this stuff. Feel free to follow along in those workbooks with some of the tests that we're going to be talking about today. And you'll notice in those PDFs, there's also data sheets for you to track some of your own outcomes on your own farm. So most of these tests do involve a few simple items. Some of them involve something you might need to buy. But most of the stuff you can just do with stuff you would have around your farm, like a shovel. So let's dive in. So the reason we're doing these, that we developed these protocols, and just as a note, the protocols are really derived from a lot of different institutions. So folks like the Soil Carbon Coalition, NRCS field protocols, Cornell protocols. So none of these are really original. So we have added some quantification approaches that are a little bit original, but for the most part, these are all tests that we didn't come up with ourselves. We just assembled them as sort of a suite of tests that would get at different aspects of soil health. So I'm going to use terms like soil health and soil carbon and soil life pretty interchangeably. And the reason for that is that these things are all really rough proxies for each other. Most soils don't contain much sort of geologic carbon. By far the biggest pool of carbon in the soil is going to be the living pool of carbon. When we increase the biodiversity of our soil, we're increasing the living things in the soil, we're increasing the health of our soil, we're increasing the physical function of our soil, we're increasing the carbon in our soil. It's all interconnected. You know, it used to be thought that soil organic matter was just like the dead stuff in the soil, right? But now we understand that soil organic matter is actually, decomposition itself is an act of living organisms, right? Because decomposition happens via biology. So when we think about soil organic matter, what we're really thinking about is a living substance. So our soil is an ecosystem. So the tests that I'm going to talk about tonight are most emerging. So, you know, by far the most common way of evaluating soil health is just a simple organic matter test. You can get that on your lab. So this is the one time I'm really going to mention a lab. Soil organic matter is about 58% carbon, and high soil organic matter usually indicates high soil health, but it doesn't always. It's important to note that there are certain exceptions, such as on an urban farm that has been recently installed, they're going to cap the native soil using gravel and you know, plastics usually, and then truck in a bunch of manufactured loam that's, you know, usually high in organic matter because it contains a lot of compost and bits of wood pulp and ground up leaves, but that might rate highly on the organic matter scale when you send it into the lab. But that doesn't necessarily mean that there's a high level of biological function in that manufactured soil. So there are a few exceptions to this rule. And remember that soil organic matter values on your lab are based on basically burning the soil and measuring loss to CO2 of that living carbon. So the soil observation that we're doing through the carbon proxy test is really about looking at soil in situ and seeing, my lamp is blinking a little bit here, I'm just tightening the bulb. Seeing what kind of emergent properties are showing up in our soil ecosystem. So what should you be looking for as a gardener or a farmer? We're not really going to be able to look directly at carbon unless we're using a soil organic matter test. But again, we're looking at the sort of living and physical indicators in the soil. So the first place we start when we're using these tests is that we choose a site to evaluate. There's two broad ways to use these tests. One is as a comparison of management practices. So say you just installed a new garden and you're trying out a no-till approach, but you have been mainly doing, and you want to see what the difference is in the new part of your garden where you're trialing no-till methods. You might start doing some of these tests on your no-till part of your garden compared to your rototilled part of your garden. Another comparison of management practices might be your annual beds that are more high disturbance and your perennial beds where you don't turn the soil very much or disturb it. Active management versus fallow. So if you have an area that's been cover cropped for a while and you want to see how the cover crop has impacted your soil. Or, you know, just your garden or field versus the fence row, just to get an idea of what the soil's like that hasn't been disturbed. Another way to use these tests is change over time. It's to conduct all of the tests or a subset of the tests on a site that is representative of your general management practices. So if you're doing that, you're going to be choosing one location and trying to find the exact same location every year and sort of stay in the same roughly 5 feet from the first place you tested, because soil is so variable. So if you're interested in looking at change over time to see how is my management practice in my field, how am I building soil life? Am I losing, you know, soil function? What change is happening over time? You really kind of want to test the same general area. So for that, for a change over time test, you want to find that location and you want to measure from a permanent landmark. And I generally just use the Compass app on my iPhone and a surveyor's tape. I have a 300-foot surveyor's tape for going out into large farm fields when I'm doing a technical consult. But if you're just in a small garden, a shorter tape will be fine. Another option is to use the Gaia GPS app, which is an app that will give you actually a fairly fine reading of your location. It's used by hikers to sort of track their hikes. But here is an example of, I recorded a little trip in a field. The red dot is the test site for this farm that I go back and visit every year in New Jersey. To do an annual test. And then I walked with the farmer to the back of the field to look at one of his other fields, and you can see our walk there. It's like a squiggly line because we walked to the back of the field and then back to the test site. So it's a handy little app if you have a lot of fields and you're a farmer and you're doing sort of spot checking and you want to do like a sample of different test locations across your field. You can use this app to record those various locations and find them again the next year. So the place that we really start in these tests, which you will have noticed if you're following along in the workbook, is surface biology. So what do you think we're looking for with surface biology and why? And maybe you, if you're looking at the grower's manual, you probably see the answers there for you, but just kind of curious about what you guys think. What are we looking for here? Feel free to come off mute. Fertility, okay, yeah, so you know how the plants are performing might tell you about the fertility of the soil. Also, what specific species are there will tell you about the relative richness of the soil. Some species that are popping up in your field, if it's weedy when you test it, might indicate leanness of soil versus, yeah, so Helen says diversity of plants and small critters. Yeah, so we're looking for sort of what's happening on the surface of the soil. Diversity of plants is an important one. With these tests, I know that this is about looking in the soil, but we start by looking at the surface of the soil because what we're really looking for is what's the plant ecology here. You know, we really want to start here because it frames the whole set of tests on, you know, the soil is interconnected with its plant community. And whether or not we're about to look at a soil that has been denuded of plants or a soil that has a bunch of rich living plants on top of it is actually going to influence the rest of the tests. And it's also going to tell us a little bit about, you know, the management practices in place at the farm. An important thing to note about these tests that I should have said at the top is that all of these tests are a snapshot in time. So that's why when we're using the test as a change over time, it's really important to take the tests about the same week every year, because all of these tests that we're going to do are going to be influenced by the time of year and the weather to a certain extent. So each one of these things is just a snapshot, and every single test might be variable based on the particular weather patterns and climate conditions of the particular growing year. But all added up, they should still tell you something from year to year when you take them all into account. This is a holistic assessment of soil health, so it's really taking a bunch of different snapshots and putting them together and saying, okay, I'm getting a picture of what's happening here. So surface biology is where we start with that. So what you want to do if you're going to be quantifying your soil health on your farm is to really record how much of your soil is covered with living plants. And how much of it is covered with just some kind of a residue or mulch. So an organic mulch like straw that you might have applied, or a leaf mulch that you might have put down, or residue like in this picture. So this is in a chestnut farm. And you'll see that in this picture, it's about 80% covered in living plants, but there's very little bare soil showing through, and the majority of what we see aside from the living plants is residue from mowing. So we would just record that as residue, and then the percentage of living plants, and then we would record the number of different species that are present on the site. So species diversity is important because it's really quite clear from the literature that the amount of different types of species that are growing in a certain plot influences how quickly and how much carbon and nitrogen are stored in the soil. So it has to do with the diversity of root types, and it also has to do with the fact that different soil dwelling organisms, different microbes, make different symbiotic relationships with different plant families. if we have diversity above, we're going to diversity below, because these encourages the biodiversity of living organisms under the soil. So living systems that are more biodiverse tend to be fuller and more full of life. So we think about the Amazon rainforest, which has so much diversity. It also supports so much life. Whereas if we have a relatively simplified ecosystem, like a cornfield in Iowa, not to shame Iowa, then we have a really simplified ecosystem and we don't support as many species. So that's why we're also looking at the number of insects present. If we have a rich diversity of insects, that's also usually an indicator that this is a biodiverse system that we're looking at. So when we're doing these tests, we're going to record them from a standing position looking down. So you're not getting down there and looking like, oh, I guess I can see some bare soil if I push over this plant. We're doing it from a standing position looking down for the canopy, leaf canopy and residue test. But then if you want to, when you're getting to the insects part, you can crouch down and kind of look a little closer and record how many different insects you see. Insects aren't the only thing you might find. You might find other stuff, which I consider a sign of earthworm activity on the surface of the soil. You might find, I have found a mouse nest on a site that I tested. What else might you find? You might find a snake skin left behind. You might find some mushrooms, all kinds of different fungi. So record all the different life that you see. If you're going to quantify this test, it's handy to just make yourself a simple observation hoop. If you have a 30-inch diameter, that's sort of the standard one that we use as a technical team, then one hand outstretched is about 6%. So you can use that to quantify like how much residue, just as a guide, like residue, living plants, stuff like that. Any questions about surface biology before I move on? Cool. So now we're going to dig a hole. Is just remove a cubic foot of soil. And this is probably the simplest and most important test you can do. It's really easy. You just need a shovel, a measuring tape, and maybe a trowel to help you. I like to bring a bin out to the field with me. Okay. So when we're looking at root behavior, we're looking at where the majority of roots terminate in the profile of the soil. We're looking to see if any of the roots are acting weird, like they're suddenly turning a corner, an abrupt corner, or if they're balling up. If you see roots that come down and then sort of form a little ball in the soil before proceeding. All of those, sort of that J rooting or sharp turn or the balling up can be an indicator that there's something, either a seasonal high water table or some compaction. We want to see roots that are proceeding straight down into the soil and are beautifully well branched. And when we dislodge some of the plant roots and hold them up, we want to see this root sheathing, which indicates a strong connection between the plant root and the soil ecosystem. You'll see that there are some roots present here that aren't well sheathed, but the majority of the roots in this image are really beautifully sheathed. It's just a lovely, we just love to see that. I really recommend looking for some grass to do this test. Certain plant families like brassicas and onions just don't form strong connections like this with their soil ecosystem, and they just won't show rhizosheathing as well. So you really want to use a grass family if you're going to be comparing different rhizosheathing. So in our technical protocols, we just sort of rank them from sort of weak to strong rhizosheathing. This would be an example of strong rhizosheathing. So we can go to the next slide. So we're looking for, well, there's two things that represented in this picture. We're looking at the presence of biopores in the soil. I also should have written on the slide that we're looking at the depth of the topsoil. I don't think that's represented elsewhere, but you can see here in this image of this really nice, nice garden soil in the Berkshires, gardener that I know in the Berkshires. She has really, really rich, dark topsoil. The roots are all looking pretty well branched. And then you can see the transition to the subsoil where it starts to change color there. one of the things that I wanted to point out about this soil is that it has a marbled appearance, going from kind of dark to light. It doesn't happen in an abrupt line. Typically when we have a soil that is like aggressively tilled, we'll see a really sharp, bright line in the soil where there's a color change. But this soil has this beautiful marbled, mottled appearance and that slow transition in a wide band. And then the biopores. In this picture, you can see several biopores in the image of the soil section. They just look like little holes where the earthworms have burrowed through the soil and left a little hole behind. And sometimes those are maybe roots that have formed a hole through the soil and then died. In this other picture of this more compacted, less friable soil, you can see there's two biopores that are really obvious above my finger on the left side of the picture there that are, I think those are from roots or really small worms. It's a little hard to tell, but those are biopores. So the presence of biopores in your soil, counting them, a quicker way is just to sort of count one wall of the soil and then times it by 4. But the biopores in the soil are an indicator of soil health. And then, of course, the other aspect of this, I think, is represented in the next slide with the earthworm count. You should get to see a little picture of a cup of worms. Yeah. So, like I said in the beginning, when we're doing this test, we want to take out exactly a cubic foot of soil. So it wants to be a foot deep and a foot, wide and long. And you want to put that into a bin, and then when you're sort of done looking at your root behavior and your topsoil depth, we want to look at, we want to sift through all that soil and count all the worms in it. It's one of the many reasons it's really important to do these tests at the same time every year is that earthworm activity in the soil is really temperature dependent. So you're going to have more earthworms in sort of the spring and fall than you will in the height of summer in July, because they're going to go deeper in the soil or into shadier parts of your garden. If you're in a garden, they're going to go hide in the shade. Generally, they travel a little bit and they want to kind of get out of the hotter soil. So definitely this is one of the main reasons that we do this at the same time every year. Generally, healthy soils will have at least 10 earthworms per cubic foot of soil in the springtime. That number, again, does go down in the summer. I was recently doing some soil tests in July, and I found 6 earthworms, and I was pretty excited. I thought that was a good number of earthworms to find in one soil in July. I have found 50 earthworms in a cubic foot of soil. That was at Noah Kellerman, one of our board members, who has a farm up in Essex County. In March, in his hoop house, under a crop of spinach, and his hoop house was built over his grandfather's barnyard. So there had been cows there for like a generation. So the soil was really deep and rich, and it was March, so it was like it was the place to be. So all of the earthworms are like hanging out in the hoop house. So that was the most earthworms I've ever found in a cubic foot of soil. I did find 27 earthworms in Julie's charred patch under a thick mulch. So earthworms really respond to being mulched. They respond to soils being moist. They respond to a lot of biological activity in the soil, because they're kind of like whales. They go through the soil and they're not just kind of digesting the dead plant matter in the soil, but they're actually digesting the living things in the soil. And they're sort of bioengineering your soil a little bit. They'll kind of like, in the gut of the worm, they're kind of sort of cultivating a little population of the beneficial bacteria that the earthworms like, and then kind of pooping it out on the other end and kind of, spreading this. They're kind of like living probiotic dispensers that just move through your soil. So, they're attracted to healthy soils conditions, and they also promote healthy soils conditions. So that's why they're a good indicator of healthy soils. So next slide. And do feel free to jump in with questions. It's not advancing for me, Jason. Are you frozen? Did I lose you all again?
No, you're here.
Okay.
I did move it to soil texture and aggregation.
Awesome. Okay. So this is kind of the hardest. This is a little bit of the hardest. Oh, I lost y'all again. Can you still hear me?
Yep, we can hear you.
Okay. It's coming back. Of the soil evaluations to kind of learn to do. It takes a lot of looking at different soil to get good at it. But essentially what we're doing is trying to understand the soil texture. And a really handy thing to have for this test is a simple little metric ruler. I like these little ones that you can slide the little guide on. And we use these in the test. You'll notice that we've classed the aggregates by their size, which is what this is helpful for. But if you look in the workbook and you print it out on 8x11 paper, you could actually just drop the soil onto the paper, and the little guides in the workbook are basically designed to give you actual size. So you can sort of use that as a guide and just sprinkle your soil on the paper if you want to just use our guide and you don't have a ruler. But generally, healthy soils will tend to have larger and more prevalent aggregates. And when I say larger, I need to qualify that a little bit by saying they're a sort of a macro aggregate is a collection of aggregates that are all sort of chunked together. But a clod in your soil or a massive chunk of soil, which I'm going to get into in a few pictures shortly, is not necessarily an aggregate. So I want to qualify the word larger. But well-aggregated soils will have plenty of pore space, and we'll have really good water and nutrient holding capacity. And again, it just takes some time to really learn what to expect in your soil aggregates, and it's a good idea to get a sense of your soils particularly. So this is why it's really helpful for you to compare different parts of your farm or garden. So look at your high disturbance areas, look at your low disturbance areas, look under your flowers in your or your asparagus in your perennial areas, look in your lawn. See if you can dig up a little soil from the edge of your driveway where your car drives a lot and notice, like, is there any soil structure there at all? So just get to know the range of the sort of soil structures on your own property, and that will start to give you a reference point. But I do want to kind of go over a few different pictures that will give us a sense, so we can advance the slide. So these are two farms that I work with in our Conservation Innovation Grant, Ironbound Farm and North Slope Farm. They're both in New Jersey. And these are examples of their soil texture from 0 to 4 inches, 4 to 8, and 8 to 12 inches. So I wonder what folks can observe about these soils. Maybe just like drop some observation in the chat or come off mute and make a comment. What are you seeing between these two different farms and at the different levels? Do you notice anything in particular?
Soil particles are finer, closer to the surface, less aggregates.
Mm-hmm. Yeah, exactly. Good observation. What else do you see? Yep, North Slope soil looks darker. Yep. What else is something you might notice about these different soil structures? What about North Slope Farm at the 8 to 12 inch horizon? What do you see there?
Moisture, water content in the soil.
Mm-hmm. Yeah, I think North Slope might be holding a little bit more moisture. You see that, so that's that chunk earlier in the slideshow. I showed you a close-up of the biopores. That's from that little chunk on the 8 to 12 inch horizon there at North Slope Farm. So that that is sort of a structure in the soil that's getting a little bit closer to what we would call massive soil. So it has less surface area than the chunk of soil that's roughly at the same part of the paper, but next to it on the 4 to 8 inch horizon. You see how there's the chunk of soil next to it in the 4 to 8 inch horizon is a little bit, has more surface area and it's darker, but the surface area is kind of key. So as we get sort of moving down the soil health spectrum, like away from really healthy to kind of less healthy, we start to see less surface area. And when we have particles of soil, or like when we break apart the soil, either with a shovel or just like breaking apart with our hands, we see more clean lines, more smooth surfaces, and less of this like really intricate surface area, which is an indicator of a lot of aggregation and soil health. Another thing to notice is that in when you compare like the iron-bound farm 0 to 4 inch horizon and the 0 to 4 inch horizon in North Slope Farm, there's a little bit more unaggregated soil, or just sort of loose soil, kind of like when we picture playground sand as sort of like the ultimate in unaggregated loose soil. We can sort of use that as a reference point and start to see when there's just kind of like, it looks like it's going to just sift through your hands, through your fingers and your hands. That's loose soil, unaggregated soil. And it's of course a spectrum. Aggregation is a spectrum from, there's like no bright line between this is like a massive soil or a platy soil and this is like a blocky aggregate. It's sort of a spectrum that we sort of try to identify. We draw a line and just say this is where that line is. It's crossed over into granular. It's crossed over into blocky, or this is platy. But we're just sort of roughly categorizing something as very dynamic, right? But generally, I think the 0 to 4 inch horizon at North Slope Farm is a little bit more aggregated than the 0 to 4 inch horizon at Ironbound Farm. But yes, they both have a certain amount of loose unaggregated soil. And on both examples, both farms, we see actually some of the best aggregation at the 4 to 8 inch horizon. And the reason for that is because both of these farms are doing a certain amount of surface disturbance. So at North Slope Farm, they're doing a certain amount of use of the power harrow, which is an implement that just kind of like egg beaters. It just kind of turns the soil in this kind of going along this way. It doesn't flip the soil, it doesn't pulverize like a rototiller, but it does disturb that top couple inches of soil, or just an inch if it's really light. So actually, at North Slope Farm, you see the best aggregation at 4 to 8 inches. And then at Ironbound Farm, we're sort of seeing kind of a slightly more even relationship between those levels. Next slide. So here are a couple more examples of some different soils. So I'm wondering if anyone has any observations about these two examples. So one thing, you know, is obviously color. We don't currently use any sort of color calibration in our tests, but there are soil health assessments that use color. The Munsell color chart is an example, but you sort of have to calibrate it for your region soils. So we haven't done that for Massachusetts or the Northeast yet. But I think recording kind of like how dark your soil is, whether you think it's dark brown, medium brown, light brown, is helpful. But yeah, here we see two different colors. We see a fair amount of surface area on the right-hand side, but I think if you look at the left-hand side, we see a little bit more surface area in that example. So next slide. Not seeing it advance. Is it on the Slake test yet?
Yes, it's there.
Great. All right. So the Slake test, if you do just one test on your farm, I would really recommend, or your garden, I would really recommend that you do a Slake test. It's really easy to set up. You essentially just get some hardware cloths. We have standardized it for, oh, I lost y'all again. Can you still hear me?
Yes, as of right now.
Right.
You might, yep.
So we've standardized it to 1/8 inch mesh hardware cloth for our purposes, but we want to have our data be comparable across different farms. So, and we're trying to quantify this, so we're using 1/8 inch. You cut a strip, you just get a 32 ounce mason jar, and you you can just set the mason jar wide mouth down on your hardware cloth and note how wide it is. So at the widest point, cut your hardware cloth strip to be that wide, and then make it long enough that you can have it fold over the sides to hang on the top of the mason jar and go into the mason jar a little bit. Fill it with water, and then take a sample of soil from your garden. We use like sort of a golf ball size as our standard, and you want to, if you're doing comparisons, take it from the same depth of different sites that you're sampling, and you can place it gently into that basket. And then what we do is we time a minute, and we note how much of the soil sample has remained in the basket after 1 minute. And then we also note again at 5 minutes. So you can go to the next slide. So we should be able to see some comparisons here of different slate tests. And we generally recommend that you take one sample A from the area that you're interested in, so your garden where you're actively growing, and then a comparison from the fence row or the forest next to the garden, or your undisturbed field next to the garden or the fence row, just as sort of or your perennial bed, just to sort of have two comparisons to see what's happening. So what are some things that you notice about these different samples? Oh, so Susan is wondering, so generally the way that we do is we take a picture right when we place the sample into the basket, and then another picture after a minute. But we also, what we're looking for are two things. One is how much of the soil sample has dropped to the bottom of the jar. And we're looking at the amount of soil in the bottom of the jar compared to the original amount of the soil in the basket, which you can still see, right? Because you can sort of see the basket and you can see the bottom of the jar. The other thing we're looking at is turbidity. So turbidity means the cloudiness of the water. So in these pictures, you'll notice that the picture of the standalone jar, the water is really pretty cloudy there. The water is much clearer on the jar to the far left. The jar to, I lost you guys again, sorry. I'm sure I'm just gonna log right back in. The middle jar is just a little bit. Can you hear me now?
Yes.
Great. So if you would like to quantify, turbidity, we played with a lot of different methods for how we could sort of try to estimate the clarity of the water, because that gets to your question, Susan, about how do you measure how much is dissolved or how much is sort of just distributed into the jar. What we're using is something called a Secchi tube. Here it is. I brought it to show you. It's this really long tube that has centimeters along the side. And, oh, I don't know if I can do this. Let's see. Can you see down at the bottom, there's a little dial, like a little pattern? I don't know if you can see that. But at the very bottom, of the jar, there is like a little black and white pattern. And this is basically what folks who do like water quality assessments for like ponds and rivers, when they're looking at like runoff and water quality in rivers, they use this simple tool, and they just record, they go out and they do a turbidity test. They're just recording the amount of centimeters of water that they pour into the tube before they can't see the pattern at the bottom of the tube anymore. So the way I conduct this test at home, I bring my soil samples home from the farm. I do the slate test, and then after 5 minutes, I take my last picture or last like note about how much of the soil has fallen through the basket, and then I gently take the basket out of the jar and place it in a container. And then I take the water, the water, which might be clear, might be murky, and I just pour it into the tube while looking down at the bottom of the tube from a standing position. And at the moment that I stop being able to see the pattern at the bottom of the tube, I crouch down and I look at where on the tube the level of the water is, and I record that. So this is just a pretty simple way to, quantify turbidity. We're trialing this as a method to evaluate soil health. Christy, great question. So the reason that we're doing these things is that we are trying to understand how well the soil, these samples of soil are resisting water causing the soil to do two things, sort of to break apart and then also to sort of more completely dissolve into the water. Dissolve is not quite the right word, but how well is the soil holding together? And can anybody tell me how well the soil holds together tells us about the soil? There's something very specific that it tells us. Anyone want to guess? Jason, can you advance to the next slide? Oh, that's just another example of a slake test. Next one. It tells us about glomalin and other bioglues. So the biological glues in the soil are really what are holding the soil particles together. It's what gives us our aggregates, and it's what causes your soil to resist water breaking apart the structure. So an important thing that, and I'm going to talk about glomalin in a second, but one of the things I want to tell you about the slake test is that when you're looking at that clod of soil in your water, and if it's like falling apart in the water, what that is telling you is that if you have a flooding event or if you have a really heavy rainstorm, which are more and more often, you're going to lose your soil structure more easily. But if it holds together really well, it's resisting the element of water, causing the structure to break apart. But glomalin is really important, and the slake test, the reason this is the one test I would say you should do if you're going to do any of these tests is because it's really important to understand how well your soil is doing in terms of glomalin and other bioglues. And glomalin is really important because it's important when we think about both your soil health and your soil structure, but it's also important in a sort of a global sense because glomalin is about 30 to 40% carbon. It's created by arbuscular mycorrhizal fungi, which are the most important organisms in your soil. And they are the most important organisms in your soil because they essentially take care of and act as sort of like the brokers for a lot of other living organisms in your soil between the plant and the soil. So you can have bacteria that will work directly with your plant roots, but if you have arbuscular mycorrhizal fungi there, for most plants, not brassicas, not certain other plant types, but for most plant families, there's arbuscular mycorrhizal fungi which will actually wrap around your plant roots and actually broker between your plant roots and a whole bunch of other soil biology. And there are whole classes of bacteria that work directly with your arbuscular mycorrhizal fungi. They're symbiotic with it. And if the arbuscular mycorrhizal fungi isn't there, then they aren't going to be there. So you have more biodiversity when you have arbuscular mycorrhizal fungi present in the soil. Glomalin is thought to account for about 27% of all the carbon in healthy soils or worldwide soils. And it can persist in the soil for 7 to 42 years, so it's a stable form of sequestered carbon. And one of the really cool things is that it excludes certain types of bacteria. It makes these, it can form these sort of filaments, these little superhighways in the soil. And this is one of the coolest things. The bacteria that are symbiotic with the fungi in your soil are going to travel along these superhighways created by tubes of glomalin, like kind of like the Roman roads, right? And exclude the bacteria that might eat those bacteria that are symbiotic with the fungi. So it essentially connects with your plant roots, and then it forms highways through the soil through which it sends its symbiotic bacteria to go to different parts of your soil. And then what those symbiotic bacteria do when they get to those different parts of your soil is that they actually start to weather the parent rock material of the soil. And so the fungi will get a signal from the plant that says, I need more, what's a good example? Silica. And the fungi says, okay, well, I've got some workers down in this corner of the fungi of my mycelium, and those little worker bacteria, I'm going to send a chemical signal to you to start extracting silica from the rock, from the parent rock material of the soil. And so the plant is paying, it's photosynthesizing, it's making, as you all learned from Jack Kittredge's talk and other talks about this, the plant's photosynthesizing, it's making carbon, it's making carbohydrates, it's paying the fungi for these nutrients, but the fungi is not making them most of the time directly itself. It's paying a bunch of bacteria to do that work. So, you know, this is this amazing system in your soil, and this is why arbuscular mycorrhizal fungi are so important. So they're important both for the fact that glomalin is really high in carbon, but they're also important because glomalin is this really important structure for allowing different biological, different biology in the soil to sort of proliferate. So I hope that made sense. And I know someone mentioned at the top of the presentation that they were interested in nutrient density. So here we have the reason why arbuscular mycorrhizal fungi in the soil benefit your nutrient density in your plants, because when you have a robust population of arbuscular mycorrhizal fungi, your soil as a living system is going to be better able and better equipped to supply your plant with the nutrients that the plant is specifically ordering up on demand. Okay? I want to tell you one more thing about glomalin. Can you, Jason, can you advance the slide? Is it showing you a picture of a mushroom now?
Yeah.
Okay, so this is, I hope a lot of you were at our summer conference last year where we had Dr. Chris Nichols as a guest, who's a big hero of mine. And she did this really interesting study. I think she was still at USDA when she did this study. But basically, she just created these little biodome environments where she and the team of researchers artificially increased the levels of the CO2 in these little biodomes, and then looked at what happened to the soil ecology in those domes within those environments. And if you're reading ahead, what you can see is that when the CO2 in some of the artificial domes had 670 parts per million. Right now we're at like 410, right? 350 is our safe limit. So they increased the CO2 concentration to 670 parts per million, which is a scary level. And when they did that, the hyphae, the arbuscular mycorrhizal fungi in the soil, responded in a really interesting way, and the hyphae grew about 3 times as much. They were increasing the body of the fungus. The mushroom is just the reproductive organ of the fungus, but most of it's the hyphae, right? The mycelium in the soil. So the mycelium got about 3 times more in volume and 3 times as long. But the hyphae the fungus then produced 5 times as much glomalin as fungus growing with, at the time of the study, what was 370 parts per million. So this is a while ago. So let that sink in for a second. When atmospheric levels of carbon were simulated at 670 parts per million, or raised to 670 parts per million, what was happening is the fungi were dramatically increasing the amount of carbon in the soil. They were ramping up their sequestration. Glomalin is really high in carbon. So the fungi were putting more glomalin, a stable form of carbon, into the soil. So I am not like a psychedelic mushroom person. I've never done mushrooms, but I definitely start to sound like kind of a mushroom fanatic when I start talking about fungi will absolutely save the world. Like, I think this is one of the most hopeful things I've learned about, and it really points to the importance of fungi on Earth as a buffer for climate change. So we can advance slide. And so this is why tillage is such an important thing to reduce globally. And so if you have a garden and you are rototilling your garden, just stop, see if you can get away without it, or reduce it. When you rototill, or when we do a really destructive types of tillage like moldboard plowing, rototilling and moldboard plowing are the two most destructive types of tillage. Tillage is fundamentally antagonistic to the physiology of mycorrhizal fungi, because when you think about how fungi exist in the soil, there's these delicate strands, right? If you've ever like turned over a wood chip pile and you've seen the beautiful little white filaments in the soil, you know what the body of fungi is like, right? So when we are constantly turning the soil and ripping it up and pulverizing it, what we're doing is resetting our fungal growth. If it's broken up, we're resetting it back to kind of like it has to kind of reset its growth. It has to start again from the remaining bits of hyphae that are present in the soil, or from spores if we've really killed it. So the other thing that happens is that when we till is that we're actually breaking up our aggregates. We're releasing a lot of sequestered forms of carbon that are protected inside of bioglue containers. We're causing a bacterial bloom for the types of bacteria that release carbon dioxide and nitrous oxide. We're causing a rapid mineralization of nutrients that make them more leachable. So we're causing nutrients that are sort of held up in the soil life matrix to just be lost with the rainfall, because it leaches into our waterways. And then some of that is being turned into a gas, right? So carbon dioxide and nitrous oxide, which we really need. We need nitrogen and carbon in the soil. When we turn it into gas and release it into the atmosphere, we're damaging our temperature control system for the Earth. So it's really important to figure out ways to tend to our soils without tillage as much as we can. And some tillage is going to be necessary for food production, but there's more minimal ways of tillage that a lot of farmers are working very hard to figure out how to do right now. So I just wanted to spend a little time talking about glomalin and tillage, and now we'll get back to our soil health assessment. That's our little interlude about glomalin and fungi.
Before we move on from glomalin, there's in the chat.
Oh yeah, thank you. So yes, clay soil that's an important thing to note. So the reason I say get to know your own soil structure really well is that different soil types are going to form aggregates and behave differently in your tests. So it's really good to do these tests on your own land as much as possible, and not necessarily compare your soils all the time to other people's soils. So yeah, so clayey soils generally stick together a little bit better, generally have better nutrient holding capacity, better moisture holding capacity. So comparing apples to apples as much as possible in your own soils. And when you do your soil test, do a texture by field procedure. It's in your technical manual. If you're comparing two different fields on your property, are you actually comparing like a more clayey soil to a more sandy soil? So you'll want to take that into account and factor that in when you're kind of evaluating how your soil is behaving. So it's a really good point. But all soils will hold together better than they would without glomalin, with the addition of glomalin. Does that make sense? And also structure. Your structure is going to be better with higher organic matter and higher life, no matter whether it's sandy or clayey. But definitely folks like me who have a really sandy soil, we have to work a lot harder for organic matter than clayey soil folks, because organic matter lasts longer and it doesn't burn up as easily in a clayey soil. And part of that is just that moisture is really important for, your soil life needs a certain amount of moisture. So if you are an excessively well-drained soil like I have in my sandy Wendell soil, if it dries out more often, it's going to be like the soil organic matter, the life doesn't hold on as well. So just a little side comment about, if you're really trying to support soil life, just kind of keep an eye on your soil moisture level. And if you're getting really dry, maybe give it a little irrigation just to kind of keep your soil microbes happy and your fungi. So infiltration, I'm just kind of looking at the time. Infiltration is another really easy test to do. I've actually never looked into whether there are commercial infiltration rings available. There probably are from like forestry supply companies, but what we do, this is my filthy infiltration ring, but this is just a simple piece of 6-inch sewer pipe. It's sharpened in one end. So I usually make a batch of these every couple of years and give them out to folks who are doing soil health assessment that I come across. But it's just 6-inch sewer pipe. I think it comes in like 40, I'm trying to remember how long, like 8 foot, yeah, 8 foot sections I think is standard, probably 16 foot sections as well. But I just kind of measure them off at I think it's 4 inches, and then use a chop saw to just come down and cut them in half, and then a diamond blade on an angle grinder to sharpen it so it gets that kind of edge. And then over time it kind of gets dented, so you can just use a simple file or a serrated knife to kind of clean up that edge. But I mean, it cost me like $40 for like an 8 foot piece of this pipe, and then you can make a whole batch of them. So if you know that you and a bunch of your friends want to start doing this assessment, you can kind of like get together and go in on it together, and someone will have a chop saw and someone will have an angle grinder. So it's really simple to make. So what you do is you just take a piece of lumber, and you get your mallet, and you get a piece of plastic, and you need a little container of that you can mark out 444 milliliters on. So this has a really rough measurement on it. So what I did is I used a cylinder that I have that has more precise measurements on it, and then I just filled it using the more precise measurement, and then I marked off the height on this little cheap plastic bottle that I carry with me in the field. And then just a piece of plastic wrap or a bread bag cut open if you want it to be really reusable. I reuse mine multiple times so I don't have to waste too much plastic. But essentially you just spread the plastic wrap over the infiltration ring after you pounded it halfway into the soil. And then you take your 444 milliliters of water and you pour it over the plastic, which is over the ring. And then you get your cell phone timer set up, or your egg timer, or whatever you have, or your watch timer. And you hit start on your timer just as you're pulling the plastic off, so you get a really precise start time. And then you just look at how many seconds, and hopefully not minutes, it takes to for 444 milliliters to drain into your soil from a 6-inch ring. And what this is doing is basically simulating a really heavy rainfall event in which we get, an inch of rain. This is basically simulating an inch of rain. That's how that's an inch of water in 444 milliliters is an inch of water in a 6-inch ring. So it's just a sort of standard way to understand how quickly is your water absorbing to your soil. So poor soil health, poor soil structure is going to cause your the water to infiltrate into your soil at a slower rate, and that might be because of hydrophobia. It could be because of crusting. It could be because you just don't have enough pore space. And generally, Gabe Brown says that a good healthy soil is going to infiltrate in under 10 seconds. I've seen that. I would amend that. In all the farms that I've looked at, generally if they have a fairly sandy soil, under 10 seconds is about right if they have other good soil health indicators. For a really healthy soil, I've seen like 3.5, 4.5 seconds. But if you have a little bit more silt or clay in your soil, then you might see more like 12, 15, 16 seconds in a still pretty healthy soil. If it's up over a minute, you're going to want to worry. It really shouldn't take over a minute for an inch of water to infiltrate into your soil. Patrick has a question. Let's see. I'll get to that at the end. So what do you think why is it important to know how quickly an inch of water is going to infiltrate into our soil? Does anyone want to tell us what that tells us about how our soils are interacting with our watershed? Take care, Susan. I guess Susan's leaving a little early. Bye, Susan. So if no one's going to hazard a guess, basically what it's telling us is in the case of like a overland flooding event or a sudden, so absorbent soils prevent flooding. Exactly. Yes, Helen, that's exactly right. I know it's simple, right? But if your soil can't absorb, if it's taking longer for water to infiltrate in the soil, then when you have a sudden heavy rainstorm, the infiltration is a rough idea of how quickly is your soil going to start having overland flooding, which causes erosion on your farm, and in extreme cases can cause soil to be washed into nearby water bodies, right? It can cause all kinds of problems on your farm or garden in terms of your soil kind of being partially washed away. You can lose your parts of your topsoil, and yes, flooding. So we've all been hearing about how much flooding is happening in the Midwest, and definitely part of that is like changing weather patterns. But I would really like to see some really good comprehensive research about how much of that is because of how poor the soil is out in the Midwest. We've gone from an average 10% soil organic matter in American agricultural soils to about 1% in the Midwest is pretty typical. So we've really lost an enormous amount of carbon out of our soils, and an enormous amount of life, and an enormous amount of physical function. And I really think that a lot of the flooding we're seeing is because these whole regions, thousands and thousands of acres, can't absorb this water. So it's running into the local waterways. It's got to go somewhere. So anyway, I think that having really healthy soils, and this is generally acknowledged by watershed associations and conservation districts, that healthy soils on farms, they hold water better, but they also infiltrate the water better. So there's two sort of separate things. Once the water's in the soil, healthy soils are going to hold on to it better. Soils won't dry out as much. But also that infiltration is a really important feature of a healthy soil, because it sort of prevents these downstream effects. Next slide. I think it just describes that. So erosion, soils that infiltrate well are more prone to erosion and protect our watersheds. Next slide. So the Penetrometer, I just want to go over briefly. I'm not going to talk as much about this because it's the thing you have to buy. They're about, where's my Penetrometer? They're about 170 bucks. You shouldn't be paying over 200. I've seen some over 200, but you should be able to get them for about 170 bucks. Generally, it's the Agritronics. I think that there may be some digital ones in being developed right now, but this is the only one I know of right now. It has this dial, which you can see on the slide there, and a point at the end. It has this is the small point. It has a thicker point for softer soils. And what you do is you push this, it has these lines along the shaft here, which are at every 3 inches. And what you do is you push this into the soil, and the dial, which I guess is kind of fuzzy in the slide. I don't know if it's any clearer in my camera here, but the dial has little numerical readings on it for the small, for the small tip and the large tip. And they start at 0 and they go up to 300. And they don't really give you a numerical reading over 300. But the reason for that is that 300 is the compaction point. But essentially what we do for our farmer clients is we take 3 readings and then we give the average reading for those farmers. So that's kind of how we track that. Because soil, again, it's variable. You might hit a rock, you might hit a little bit of a hard spot in the soil or a particularly loose spot. So an average is kind of nice. Let's go on to the next, I lost you guys again. Let's go on to the next slide, Jason. Just to give you an example of some different readings that we've taken.
Okay. Hold on, Caro, you're muted somehow.
Sorry, can you hear me now?
Yep.
So basically any plant that we're growing as a crop can't penetrate soils where the PSI is higher than 300. So this little chart just gives you a rough idea of kind of the percentage of plant roots that can penetrate at the pressure that you're finding, the PSI, the pounds per square inch. We have on the bottom here 2 farms that I've recorded data for, just an example of 2 extremes. We have a tilled grain farm on the left, and on the right we have a no-till vegetable farm. And you can see how dramatically different these readings are. They're both in the same time of year. So soil moisture does influence these readings a little bit. But you can see how quickly, so at 15 inches there, we get to over 300 pounds per square inch of pressure needed to push the penetrometer through. So he's got a plow pan or a compaction level at 15 inches. So his roots, his crop has only 15 inches of soil to play with. Whereas this other farm, the no-till veg farm, has really loose, light soil way, way down. They're on a former alluvial plain, so they've got organic matter way down. It was like a wetland like thousands of years ago. So their plants could just send roots on till daylight, you know. So it's a handy tool to have. If you're really serious about your soil health, I recommend picking one up and going around your property and just poking it around and seeing what you find. It'll tell you a lot. It's a really useful tool, but it's a little pricey. Next slide, Jason. Last thing I wanted to mention is also a thing you have to buy, but it's really cool. Unless you're a chemist, a home chemist, and you want to make your own reagent, there's instructions for doing that online. If you just look up potassium permanganate solution for active carbon test. But you can go to Soil One. There should be a website provided in the materials page of your handbook in your folder. And you can buy a simple field kit or a more professional kit. And essentially, this is just a test where you look at how much exchangeable carbon is present in your soil. So exchangeable carbon is just like the carbohydrates and the root exudates and like the bits of carbon that are being digested by one microbe, and like then the microbe gets eaten by something else and it becomes part of that microbe's body. It's really the carbon that's being exchanged around in your soil ecosystem. And what's that question? I thought I heard a question. So this is just a really, it's just a helpful little test for kind of understanding how much activity, biological activity is present in your soil. And I think I'm just going to leave it there. Oh, sorry, just the, your result is the potassium permanganate is dark purple, and it reacts with the active carbon in your soil, and it will get really clear in response to how much active carbon is in the soil. So in this example here, in the picture, the dark soil, the dark purple is a low organic matter, low biological activity soil, and the really excellent soil is the clear, it looks like water. So I think I have a couple left, 3 minutes for questions. Any final questions? I know that there was one in the chat earlier, Jason, but I lost it because I got kicked off.
Yeah.
Can you just read that one to me? Yeah.
Yeah, sure thing. Patrick says, I understand the need to eliminate tillage, but when converting forests with large 4-foot diameter trees, that's 4 foot, right? 4-foot diameter trees to soil that can be easily formed, seeded or transplanted into, it seems large amount of soil disturbances required. Is it correct to assume this is unavoidable and that soil health can then be restored?
Yeah, that's such a great question. I'm spending a lot of time thinking about stuff like that because I serve as a project advisor for something called the Massachusetts Healthy Soils Action Plan. So I'm working with a big team of academics and practitioners and folks who are putting together a report for the Executive Office of Energy and Environmental Affairs about how Massachusetts should make sure that the state has more healthy soils. And one of the big findings of this is really the best use of land in Massachusetts is forests. If we're thinking about soil carbon and greenhouse gas sequestration and soil health broadly, forests in the Northeast have around seems like from what I can find, like 6 to 8% organic matter, and the average soil organic matter for farms and agricultural soils in Massachusetts is like 2 to 3%. So yeah, when you convert a forest to gardens or ag land, you're really all that soil disturbance is going to take that organic matter right down. And we do see that, typically when you convert, I think there's a good statistic in the carbon farming solution by Eric Tonsmeyer. I think it's that agricultural soils lose about half of their carbon after being managed in agriculture for 50 years, I think it is, in the Northeast, and it's like 10 years in the tropics. But yeah, if we're thinking just in terms of carbon, then forests are the best use. And logging does, there are best management practices that can mitigate the impacts on soil health for logging, but it is a disturbance event, period, and it does have an impact. But then, there's issues of like maximizing biodiversity in forests. Sometimes the forest does need to be managed because it's got a certain kind of species dominance because it was planted for a certain type of softwood plantation, for instance, and the biodiversity is reduced. So that forest might not actually be reaching its fullest carbon potential because biodiversity is also really important for maximizing our carbon. So I don't know, that's not maybe a great full answer, but I hope I got at it from a couple angles.
Yeah, thank you.
Christy, how much is too much? What percentage of organic matter do you have in your soil there? I see your question. My recent UMass soil tests have been telling me I have too much soil organic matter. It seems like it should be a good thing, but I understand from some investigation that can be a problem. Do you know what the level is?
I don't have the test right handy, Caro. So I don't recall what it was. It's 2 years in a row now. I've been getting that result, and their advice was don't add any more compost or organic material. And I can't get my head around why that would be a problem.
Yeah, is that Christy Higginbottom?
Yes.
Hi, Christy.
Hi.
Yeah, why don't you send me your soil test? I don't want to get in trouble here.
Okay. Would something like that impact seed germination? I've noticed all of a sudden I am getting a very poor seed germination, something I've never had before.
Okay, yeah. So I've noticed the same thing with my soil. Yeah, so hydrophobia can be a problem. I guess the one problem with too much organic matter might be hydrophobia, in which case you might actually need to disturb your soil a little bit, which is sort of counterproductive to everything else we've said. Not disturbing your soil is going to help you build up soil organic matter, but if you have been adding a lot of compost and you're starting to have germination issues, you might actually need to work that organic matter a little deeper into your soil. So gently doing that with sort of as little pulverizing of your aggregates as possible. So maybe taking like a garden fork and gently turning it over lightly might help you there. I got kicked off Zoom, but I'll keep talking. But so if you have too much organic matter right on the surface of the soil, you might be having issues where it's so much organic matter that it's drying out because it's so light. So that I have seen that issue before.
Thank you.
The other thing, one more thing you can do is if you do need to add any amendments, switch to a clay-based amendment like soft rock phosphate. What's another one?
My tests have also been telling me I have too much phosphate.
Yeah, okay, so don't use soft rock phosphate. Spanish River carbonatite and aragonite for calcium. Those sort of clay-based amendments will help with that a little bit too. But yeah, generally I don't know. I think that at NOFA we recommend a higher level of soil organic matter than the extensions are going to recommend.
That's what I thought, maybe.
Yeah.
Caro, there's a question from someone saying, I see so many spiders in my garden. Any idea what this means?
I'm sure that's a great thing. So if you have a lot of spiders in your garden.
In my garden soil, Oh,
your garden soil. So like they're like the kind of spiders that make a little hole and then they like pop out. What do you mean in the soil?
Yeah.
I think so. Okay. Yeah. I mean, so like if you think about a healthy ocean has a lot of whales, right? That means that's an indicator that there's a lot of fish in the ocean. It's the same thing with like the apex predators in your soil, right? So like if you have a lot of spiders, that probably means that there's lots of critters for them to eat. Yeah, so I don't think that that's a problem. Sounds like it's a good thing. Although it could mean that your soil's maybe a little too dry. I don't know. If your soil's really dry, you might have spiders moving in, but I don't know. I have to think about that. The best crop to grow excess P and K out of the soil. What do you mean? To try to remove it? You really there's not really a way to do that unless you're kind of growing a bunch of cover crops and then like cutting them and moving them off your field. I don't really know. I don't think you can really address. The only way you can really address too much of an element is to try to balance the other elements and bring the other elements up. So if you're looking to balance out your P and K with your calcium, you know, at NOFA we recommend a calcium base saturation of between 65 and 75%. If you're looking for recommendations on amendments that will help you balance your phosphorus and potassium, you can send your soil test to Laura Davis at nofamass.org. I'll put the email. Laura Davis at nofamass.org. And if you're a member of NOFA, it's just $25 for her to read your soil test, interpret it, and send you a specific mineral amendment recommendation to balance your cations. And also she'll give you, if you get trace mineral analysis on your test, she'll also give you which trace minerals to put in. So she's our soil lab analyzer. She's really great. But I can't really tell you. I don't know a lot about the chemistry. I just know about the biology. All right. I want to be respectful of people's time. I know we stayed a little late because I kept getting booted off. But any final last questions before we let everybody go do something else with their life? Thank you so much for your patience with me, folks. I'm so sorry. I came down to this farm to try to have a better internet connection, and it hasn't been working out for me today. So I really appreciate your kind forbearance and your patience with my issues.
Thank you very much, Caro, for bearing with it yourself, and for everyone.
Thanks, Jason.
Yeah.
Thanks for hosting.
My pleasure. My pleasure. Yeah, and thanks everyone for hanging in. Really appreciate that. It's great to see you.